All right, good morning, everyone. Thank you so much for coming to our first webinar of the year. So happy 2025. Welcome to the new year. I'm very excited to have our speaker today, who is another one of the GBatNet student scholars, Lexi Frank, and who is a PhD student at the University of Minnesota. But before we get started, a little bit of housekeeping. Just a reminder that we do have our webinars at the first Wednesday of every month. So the next one, I think, is February 5th. It's going to be the first Wednesday of February. So be on the lookout for our next speaker. And yeah, I think that was probably about it. But we can go ahead and get started. So again, we are going to have Lexi Frank, who is going to be our speaker today, and she is going to be taking us through kind of a story of netting bats and identifying them based off of their DNA right there in the field. And if you wanted to follow along for the step-by-step process, go ahead and download the link that was attached in the email. It has the code that we're going to be using, and we can follow along. So without further ado, Lexi, take it away.
Sweet. All right. Thank you for the introduction. I'm super excited to talk about the subject. It's really fun to me, and I'm hoping it'll be fun for you guys. So, we're gonna learn about using DNA to identify species in the field, and we're gonna go through some bioinformatics and hopefully be able to together identify this mystery bat right here in the corner. Okay, so just a little bit brief background about me. Dana already mentioned I'm a PhD candidate at University of Minnesota. I'm also a GBatNet student, and I mainly am studying small mammals, so mostly bats, but also a little bit of small rodents. And then thinking about disease, genomics, metagenomics, epigenomics, and then working with a mobile laboratory that we're taking in the field to do molecular work. And I do most of my field work, well, I do some in the US and also some internationally. So today I'm going to take you along on some field research in at one of my field sites in Guyana in South America. And so kind of our agenda is to travel to our little field site, catch some bats together, then we'll extract and sequence our DNA, and then we'll be able to analyze those sequences bioinformatically together and hopefully be able to identify our species. And so this workshop, I sort of pulled together a bunch of different data and situations from the field. So it's all, it's a pretend field trip, but it is based on some real stuff. So in Guyana, I've been to a few different field sites to do a collection of bat samples. The stars are different field sites I've been to, and then the black dot is Georgetown, the capital of Guyana. So that's usually where we'll fly into from the US. And so today we're going to be going all the way to south of Guyana to the Orange Star. And so this orange star is at the Conishan Amerindian Protected Area. So this is a region and a protected area that is owned and managed by an indigenous community called the Wai. And their main village is called Masakanari. And there's a picture of their Benab, which is a community center right here at the bottom. So this protected area is really unique and really a great place to work because there is only a population of about 300 people, and there's about 625,000 hectares of protected forest surrounding this area. And so that is really awesome to have that protected forest there for bat diversity and uh, it's a great place to work also because of the people that are there. They're super interested in conservation and zoonotic disease monitoring within their protected area. And in general, there's pretty low levels of human disturbance because mainly the YY are doing subsistence hunting and farming. And so I'm super lucky to be able to work at this site and it's been possible because of Dr. Chris Schaefer and Dr. Marissa Milstein. They established a research connection with the Indigenous YY, and so we're able to work with them on goals that they're interested in that also align with our research goals. So that's where we're going today. So we are flying into Georgetown at the Black Dot, and we're going to travel all the way down to the Orange Star. And so also within Guyana and within this region of South America, there's really high species richness. So there's over 130 bat species recorded in Guyana across 9 different families. And then there is a lot of cryptic species, so species that have low to no obvious morphological differences, but are considered different species. And then we've also got a lot of species complexes going on. So these are organisms that are closely related and really similar in appearance, but we're not necessarily sure what the phylogenetic relationships are between them. So we're traveling to our field site. So we're starting off in this minibus in Georgetown, and we're starting the drive all the way down the length of the country. And so these minibuses are great, they're strong, but they're not necessarily anything compared to these roads. So immediately we're stuck and we're pulling ourselves out. And then we think, okay, we're out, great. But then we're stuck again and we're pulling ourselves out again. And we think, well, the roads can't possibly get worse, can they? But they do, in fact, get worse. And so we've got all of our stuff in this van. We're working hard, we're pulling ourselves out, but it's slow going. So eventually we have to get some heavy machinery to pull us out. And then we switch vehicles. But unfortunately, our vehicle breaks down. So we've got to get it fixed. We think it's fixed, but turns out we actually need a tow. We get towed by this truck for hours and hours. But finally, we think it's fine, but it's actually not, and we're stuck again. This time, not just in mud, but also in a huge puddle. And then we're stuck one more time. We think maybe that's it. We're almost there. Nope. Got to get pulled up by a tractor again. And then one more time. Stuck again. But we finally make it to the river where we pull out all of our equipment, put it on a canoe, and this is like a super long canoe. We've got all of our, over 1,000 pounds of equipment on this canoe, and we all get in there. All, however many of us are on this call are in this canoe, and it's got a motor, and we motor down the river for a few days to get to our final site, which is Masakanari Village. So this is a super awesome community up on a hill, just off the river. And we finally made it. So we're gonna catch some bats. So I've set up a bunch of different little icons here, and actually, if anyone wants to unmute and say where we should put our mist net, you can certainly do that. But if not, I can also pick some. So let's see. So if we set up a mist net over by this chicken coop, we're gonna catch this little bat here. So also, if anyone has questions throughout this, you can fully feel free to unmute and just yell it out or put it in the chat. Um, but yeah, I want it to be interactive. But yeah, so we've caught this bat, and this is a bat that has a forearm of about 54 millimeters, and so forearm length is often used as a distinguishing feature. It's got compact muzzle, large thumbs, and so we're able to identify this as Desmodus rotundus. So this is a blood-feeding bat. And then we're gonna put up some more mist nets, maybe, let's see, over here by the river. And we've caught this little guy. So this is a much smaller bat. It's grizzled brown and has 2 faint stripes on its back. It's also got some evenly spaced tufts along its forearm here. And so we're able to identify this bat as Rhynchonicterus naso. So this is a bat that like lines up on trees near rivers. And so now we're just hanging out. We're not catching bats, and we decide to go over to the pit toilet, to the latrine, to do our business. But actually, there's also bats in there. And lucky enough for us, we like bats, so we're excited to see it. There's a whole little roost of some Carolia in here. And so Carolia are super common bats in this area, especially near communities of humans. but they do have some species that have overlapping similarities. So the Corolla perspicillata and the Corolla brevicata actually have some overlapping measurements in forearm and tibia lengths. So these are often a good candidate for molecular field ID if you really need to know the species. But that's not our mystery bat. So now we're going over to this forested area and we've caught our mystery bat. So we don't know what this bat is based on the features, and I'm not going to show you the picture of it quite yet because I don't want anyone to spoil it and guess what it is yet. But we've got a forearm of about 63 millimeters and a weight of about 24 grams. And we captured it near the forest and near humans, but also near the river. So there's a lot of different options of maybe what the diet of this bat might be. Oops. So thinking about molecular ID in the field. So why do we want to do molecular ID when we're in the field specifically? And so again, going back to thinking about cryptic species and species complexes, sometimes it's not very clear just by morphology what bat we're looking at. And there also might be certain changes in taxonomy that have happened recently to either split or lump certain species. And so we might not even be aware in our field guides what has been recently changed. Also, sometimes you actually do just need the full-on skull for that morphological ID, which you might not be able to get, in the time of your study at your study site. And then sometimes you're working with a field guide or a key that is from a geographically distant place from where you're actually working. So it might be similar bat species, but it's far enough away that there are differences. And then sometimes it's just super important that you get that actual confirmed species ID. Maybe you're doing a study about disease transmission, or maybe you're working in industry and it's really important to your clients to know the exact species ID. And then finally, another reason why we might do our molecular ID in the field is because you might have difficulties moving your samples from your field site to the lab. And that could be due to costs. It could be due to, you have sensitive samples and you have to travel a long way. Maybe something happens, the samples aren't preserved correctly, or they're not preserved in a way that, accounts for every single thing that could happen. It also, often takes a long time to transfer your samples from your field site to the lab, and maybe you have deadlines, so you need to have data to be able to work with before that. And then another big thing is local, regional, countrywide laws and policies may change or be different, or you didn't know, and so it may delay your ability to transfer from your field site to your lab. But again, molecular ID in the field, it isn't always necessary. So you really have to pick and choose, like, is this worth the time and resources to be doing a molecular ID in the field versus getting your samples back to your lab, wherever that may be. So when we're doing species ID in the field, we're often using the mitochondrial genome. And so the mitochondrial genome is super great for species ID because it has what is called barcoding genes within it. So it has genes like cytochrome B and cytochrome oxidase 1 that are known as barcoding genes. So they help us identify species. And this is because there is a known rate of evolution in these genes, so we can tell by how much they've changed from each other when when a different species is present. And it's also great for sequencing because within a cell, you'll have your nuclear genome and you'll have, a singular copy of that, or two if you want to call it. But the mitochondrial genome is actually present in each cell in between 100 and 10,000 copies. So you have a ton of copies of this mitochondrial genome, which is great for sequencing. And it's also a relatively small genome. It's about 16,000 to 17,000 bases. So when we're sequencing, we can't just use any sequencing technology. We have to specifically use a portable technology when we're in the field. And so nanopore sequencing is that portable technology. And so the sequencer is here in the center. And in real life, it's about the size of a king-size candy bar. So you can really take it with you when you're going in the field. And then it works by here's a enlarged version of a nanopore. So this is the pore, and then this is your DNA, and the DNA is pulled through the pore, and as it goes through, the computer and the sequencer is able to say what each base is. And so it's super awesome because it's portable, it's affordable for individual labs. It's about 2,000 US dollars for a sequencer these days. And that is really cheap compared to like an Illumina sequencer, which is hundreds of thousands of dollars, very not portable. So this technology is really great for the field. And you can also, as you're generating data, like as you're sequencing, you can start analyzing your data like even while you're still sequencing. There's also no length restriction of how long of a fragment you can sequence. So that means however long this DNA piece is that's going through the pore, you can sequence that whole thing. And that's super great for genome assembly or assembling, longer like pieces of DNA or longer genomes or anything compared to short reads, which are much harder to do assembly with and also harder to assemble certain regions of the genome, especially like repetitive regions. And also in general, library prep, so preparing your DNA to be sequenced is generally pretty straightforward. If you have, some experience pipetting, you should be able to pick it up pretty easily. So when we're doing our field IDs, we're using the mitochondrial genome, we're using nanopore sequencing, but we also want to use a technique called adaptive sampling. So adaptive sampling is really cool because it helps us to be able to enrich for the type of DNA we're looking for. So in this case, we're enriching for the mitochondrial genome. And so adaptive sampling works by you give the Nanopore software a file that has all of the sequences that you're interested in trying to enrich for. So we would give it a file of maybe like all published mitochondrial genomes of, mammals. And these can be published on NCBI, so that's the national sort of repository for this type of sequencing data. And we can go in and we can say, let's download all mammal mitochondrial genomes that are published on this website. And then we can give that to our nanopore software. And then as we're sequencing our DNA, so as a little strand is going through the pore, the first 200 to 400 bases will go through, and then they'll compare those bases to our reference, so to our downloaded mitochondrial genomes, and say, is it a 70% match? And if it is above a 70% match, it will keep sequencing the rest of the piece of DNA. But if it's less than a 70% match, it will kick that piece of DNA out of the pore. So we're not sequencing that. And then basically we're enriching for mitochondrial DNA. So if any nuclear DNA is coming in, we're not getting that. We're kicking that out. Any like contamination or bacteria, we're not doing that. We're kicking that out. So we're able to get more sequences from mitochondrial genomes, which is great for assembly because having more sequences will lead to higher confidence in our ID. So we've got our bat of interest. We don't know what it is. And now we need to set up our mobile lab. So we are really lucky. We've got some super awesome people on our team from the local YY community, and they are able to set up a nice table for us, help us. Like honestly, they took care of us so well, and they were super awesome in helping with the bat work as well. And so we've set up our mobile lab. And some of the equipment that is super important to have is we need some pipettes, we need a heat block, as well as a centrifuge. The magnetic rack is important for nanopore sequencing because you use some magnetic beads to help separate DNA. And then a qubit is a way to quantify how much DNA is in your sample. And then over here in this picture, we also need our laptop and a generator or some sort of power source. You can also do this with solar power, with just like regular plug-in wall power, or a large battery. And then we've got a cooler to keep our reagents cold because we are in a neotropical region. And then we finally have got our nanopore sequencer over here, which you can barely see in the picture because it's actually so small. Um, so that's our setup. And now we're gonna do our DNA extraction. So you can choose your sample type. So you can either do, like a muscle sample, liver, kidney, like any tissue you can use, but you can also use some non-invasive or non-terminally collected samples like fecal samples. You can use a wing punch, and you can use even blood. So anything that has cells that has DNA can be used for this. And so these are some basic steps of DNA extraction, but you would typically homogenize that tissue, so break it up a bit. You would lyse open your cells to release the DNA from inside of the cell. And then you would separate that DNA from everything else that makes up the cell. So separating the DNA from all the other proteins and enzymes and lipids and everything. And then finally, you'll have your precipitated DNA just in solution here by itself. And so again, the great thing about nanopore is you're working with however long your DNA fragments are, you should be able to sequence, the whole thing. And so when you're assembling a genome like a mitochondrial genome, it's nice to have these big long reads because where they overlap will help us to assemble our genome. And so these long reads are great for getting that overlap. Whereas short reads definitely have a purpose and they have studies that they are great for. But for assembling genomes, they're not quite as good as long reads because you're often left with some gaps where you are not sure what is in these gaps, but you can assemble these other sections. So now we're going to go into our data analysis. We've done our sequencing. We caught our bat, did our sequencing, and now we're going into our bioinformatic analysis. So what you're going to need for this specific analysis, I do it in command line. So you need access to the command line, and I'll show what that is in a minute. But normally, I'm doing it on either a Linux or a Mac computer. There's also a website called Galaxy where you can access a lot of these tools just through the website, and you can upload your own data and use pretty much all of these tools. So that's also a great website, especially if you're a beginner. So then our sequence data is stored in what's called a FASTQ file. And so we also will need all of our sequenced FASTQ files. And then finally, we're gonna use those references, those reference mitochondrial genomes that we downloaded from NCBI. And then some of the packages we're gonna use, Nanofilt will help us filter our data. Minimap2 will help us align or match up our reads or sequences to the reference. SAMtools is a really versatile tool that you can use for sort of manipulating, like indexing or viewing, editing your data. And then Fly is actually what is the program that will assemble our genome. And then these other two, so within Galaxy, you can use a program called Mitos2, and that can help you annotate or label the genes within your genome. And then RaxML is for phylogenetic tree construction, but we probably won't make it to these two steps today. But I do have those included in the HTML file that was provided, so you should be able to go through those steps later if you'd like. So I'm going to pop out of my slides here. Oops. And go to, so here I just have open that same HTML file that that Dana sent out in the email. Okay, so in this file, this is basically, I've just edited it for this workshop, but it is originally from this paper that I published with my colleagues last year that goes through the same process and just uses fecal samples specifically. And then I've included first some basic commands in Linux. So I'm on a Mac right now. And so when you're on a Mac, you can access the command line or the terminal by going to this little icon. It's called terminal. And if you click it open, you get some words here. So this is basically just a text-based way to interact with your computer. So this is just a way that I can manipulate files for this process in a specific way and use packages. But this is all just on my computer. So the first command here is called cd, so that means change directory. So if I put cd and I say desktop, that will take me into my desktop. And so here you can see it says Larson Field. That's the name of the account I'm on because I'm in Peter Larson's lab, and this is our field laptop. And then at Padme, Padme is the name of the computer. It's a bit of a Star Wars theme around here. And then now you can see we're in desktop. So if I say ls, which is the next command here, list, we can list out the different directories or folders that are in our desktop. So we've got data, data, logs, files. And if you go over here to look at the desktop, those are the exact files or directories that are visible on my desktop. Okay, so then I'm going to change directory or CD into my data folder because that's where I've stored the data for this project. And then I'll list again. And so I've got several different things in this folder. So I've got a readme.txt file. So this is just a text file that basically says what everything within this folder is. And I would say if you learn one thing from this, it's that documentation is super important in bioinformatics because as I'm typing these commands, I'm typing them, but if I don't label my files that I'm creating in a certain way, or if I don't record what I've been doing, it can very easily be forgotten, and then you won't be able to recreate it. And I'm telling you this out of experience because this has happened to me. Um, but then I will CD into my experiment folder. And so this is another folder where I should have my data and I'll list. And so in here, I've got barcode 9, barcode 10, barcode 11, and barcode 12. And so each of these barcodes is actually representing a separate sample. So when we did our sequencing, we can put little tags on each of our samples to say, like, this is bat number 1, and this is bat number 2, and then so on and so forth, and bat 9, and bat 10, and 11, and 12. So each of these is data for a specific bat. And the bat we're working with, our mystery bat, is barcode 10. So I'm going to CD change directory into barcode 10. So now we're in there. Okay, so I'm going to list the contents. All right, so we've got a ton of files. It's probably looking pretty overwhelming right now, but they're all named in a way that is easy to understand once you get what it is. So all of this is just information about the run, but it all says barcode 10, so we all know it's for our specific bat. And they're all these .fastq files. And so those again are files that are storing our sequencing information. So if we use the command head, so head will show us the first bit of our file. So we're going to look at the first bit of one of these files. Okay, so press enter. All right, so again, this looks pretty overwhelming, but it is in a certain format that we can understand. So this first bit, first 3 lines are all metadata about our sequencing read. So it will tell us like the time it happened, what version of technology we were using, and then the barcode. So this is all great information. Then we've got our actual sequence. So these are our C's, G's, A's, and T's from our DNA. So this is our literal representation of our fragment of DNA. And then you've got this plus sign, and then you've got some other kind of crazy symbols and everything. And so this section is quality information about our read. So this section is not intended to be human readable. It's read by your computer and understood by your computer, but you can't really read it yourself right now, as you can tell. But this tells us basically how confident we are that each of these bases in here is what it is written as. So how confident are we that it starts with a C and then has another C and another A and A? Okay, so that's the FASTQ format. So then if you do the command clear down here, it will clear out so you can start fresh on your screen. So now we'll head in to the actual analysis. So the first thing, navigate to the directory or folder with your sequence data, so your FASTQs. There's also a different type of file called a FASTA, and so that just has only your sequencing information. Instead of having the metadata, the sequencing information, the quality information, a FASTA just has the name and then the sequence. But we're working with FASTQs today. So we're already in our correct directory. So now we're going to, here, I'll list again. Those are all of our files. So all of these have reads. They have a bunch of reads from barcode 10. So the first thing we want to do is we want to put all of these FASTQs together into one file so that we can work with just one file that has all of our barcode 10 reads. And so within this document, it talks about doing a G unzip. But as you can see here, I already have unzipped the files because that does take a minute and I didn't want to waste time doing that. But if your files are zipped, you would just use this command. But we don't need to do that today. So we're going to use the command cat. So that means cat can either be used to look at the contents of a file, or it can be used to put files together. So to concatenate them or put them together. So we're going to cat. We're going to use this little asterisk, and that just means any character. And then we're going to say dot fastq. So this will mean put together any file that is a dot fastq in this directory. And then we are going to call our output file all barcode 10.fastq. And so it's important to name it in a way that will help us understand what is in that file. So we're saying everything from barcode 10 is in this fastq file. Okay, so we'll run that, and it's pretty quick. Then we'll say ls to list to make sure that it created that file. So now we've got the file here, all barcode 10.fastq. Okay, so now, next step, we're using a program called nanofilt. And so each of these programs I have linked in the HTML, so you can click the link and it should tell you how you can download and use the programs. It's often pretty easy, and there's usually like a place where people have, put if they need help or what problems they're having. So you'll just have to download that software to your system. And then Nanofilt is a program that helps us filter our sequences. So first, if we look at it, we have our nanofilt command, then we've got a dash and the letter Q. So a dash and then a letter is called a flag. And a flag is basically a way that you can say specific things or specific features that you want to happen with your software that you're using. So the nanofilt filters nanopore sequences. This dash Q stands for the quality score you want to filter for. So we don't want any sequences that are under a quality 7. And so a quality 7, it's sort of it's not, super great of a quality. 10 would be a lot better, but we just want to get rid of everything that's super bad. So that's why we're doing minimum quality 7. Then the dash L means what is the minimum length that we want in our file. And so we're going to say the minimum length that we want to see is a length of 300 bases. So we don't want any fragments, any reads of DNA that are less than 300 bases. And this is because, again, we want those longer reads. They're better for assembly, and some of the shorter reads can be kind of junky, and they're not great for our assembly. So then we're saying that we want our max length to be 17,000. And so that's because we know that the mitochondrial genome has a length between 16 and 17,000 bases. And so we don't want anything longer than that because that would mean it's not from the mitochondrial genome. Then we will give it the file we want. So that file we just made, the all barcode 10.fastq, that's the file we want to filter. And then we're calling it, the output, we're calling all barcode 10, and then we're saying Q7 300 17K. So that kind of helps us remember what were the the different parameters that we put into our filtering. So then we'll run that, and that should also be pretty quick. And then if you ever are using a program and you don't know what the flags are, you can always say nanofilt and then do a -H, and then that will pull up basically a help menu. And so this says, -H means help, -V would mean give you version information. It says the -L is your minimum read length, so you can get all this information and decide how you want to use it. So let's do a clear. So we'll list, and we'll see that we created this file, all barcode 10 Q7 300. So we created our file, and we can check that by using a program called SeqKit stats. We can first pull up the stats for our original file. So here we've got some stats for our original file, and then we can do the same thing, SeqKit stats, for our new file that we filtered. So we can see that both in FASTQ format, both DNA, this original file had 195,000 sequences, and then now that we've filtered, we have 176,000. So that's really good. That means we filtered, we lost sequences, which is what we were intending. And we also can see our minimum length went from 55 bases to 300. So that's all good. That means it worked. So we'll clear again. Okay, now we're gonna map our reads to our reference reads. So we're mapping the reads that we produced to our reference reads. And so I've already made the reference here. And then if you go to my original paper that I published, linked at the top of the HTML here, there is information in a HTML folder in the supplementary data about how to create that reference folder or file if you need to. But basically this reference here has all mammal mitochondrial genomes that are of reference level quality, so really high quality mitochondrial genomes from NCBI. So we're using Minimap2 to do our alignment. So the -AX is another flag that's specific for nanopore. Map ONT, so ONT stands for Oxford Nanopore Technology. So this is for nanopore reads specifically. Then we've got our reference, and we've got the filtered file that we just used. And then we're going to name it again. So we're going to name it the output, all barcode 10, and I'm calling it mito reads. And now we're changing the file type. So we're going from a FASTQ to a .sam file. And so a SAM file is an alignment file. So it's a file that tells you your sequence, but also where it aligned to your reference. So we're going to run that. And that sometimes takes a second, but it shouldn't be too long. Okay, so it's run. Let's list and make sure that we got our new file. So yep, we've got all barcode 10 mitoreads.sam. So now we're going to use SAM tools over here, and we're going to be able to manipulate that SAM file a little bit. So the first thing we're going to do is we're going to use SAM tools view. So SAM tools view is going to help us change our file from a SAM to a .bam file. So the difference between SAM and BAM is not much. It's the same file. It's just that a SAM you can read as a human, but a .bam file, the B stands for binary. So we're switching from a human readable file to a binary file. And we're doing this because binary files are smaller. You're able to work with them more quickly because they are smaller, and it's easier for the computer to use them. So a BAM file is often the type of alignment file that you would be working with for your analyses. So we'll go ahead and do that. The next command we're going to do is we're going to use SAMTools sort, and that's going to sort our BAM file, so our file that has all of our reads and where they're aligned to in our reference, and we're going to sort it so that it's in the order of the mitochondrial genome. So we want it to follow the same order that the genes have in a mitochondrial genome. So first we'll check for that last file. All right, we've got our BAM file here created. So now we're going to sort that BAM file we just made, and we're going to output it as a new BAM file. So we're still working with the BAM. So now we're sorting it to the correct order within the mitochondrial genome. And that usually just helps your analyses further down just go a little bit faster. So next, we're going to use SAMTools view, and we're going to pull out all of the reads that we're mapping specifically to our reference. So right now in our BAM file, we have all of our sequences, how they mapped, but we also have the unmapped sequences still in there. And we want to get rid of those and take them out of our file because we're not interested in anything that didn't map to the mammal mitochondrial genomes. So that's what these flags here are standing for. They're saying take out all files or all sequences that did not map to anything at all. And we're going to use the BAM file, the sorted BAM file that we just created, and we're going to call it in the end all barcode 10 mapped.bam. So we'll run that. That one's pretty quick. List to make sure it's there. So we're listing it and we're seeing all barcode 10 mapped.bam. So got that one now. Okay, and then our final step with SAMTools is we're going to turn our BAM back into a FASTQ. So we're using SAMTools BAM to FASTQ. So we're taking our all barcode 10 mapped.bam and we want the output to be called all barcode 10 mapped.fastq. So we'll run that. And then you can see it gives you a little output and it says it processed about 2,443 reads. So that's really good. That means we've got reads and we're ready to assemble them into our mitochondrial genome. So now we're going to switch to our next program. So this program is called Fly. And so Fly is an assembler. So that means it will help us assemble our reads into a full genome. So we'll use Fly and we're going to use --nanocore. So that just means it's 4 nanopore reads that we're using. We're going to give it our all barcode 10 mapped.fastq. So all of our reads that were mapped to our reference file. Then we're going to say --genome size, and that our mitochondrial genome size is about 16K, so 16,000 bases. So we'll give it that size. And then it's going to output a directory or a folder, and we're going to call that folder assembly underscore barcode 10. And so it'll put all of the files that it produces while it's assembling our genome into this file or directory. So we'll run that, and that gives you some more output, and it'll take a second here, but not too long, hopefully. And, you know, one of the great things about using the mitochondrial genome is it doesn't take too long to run any of these programs. If you used like the full nuclear genome or you're trying to, assemble a full genome in the field, it would take a lot more computer power, a lot more time. But the mitochondrial genome is quick. Okay, so now let's clear first, and then we'll list out again the contents. So we've got all the files we generated, all of our original FASTQs, and we're going up here. And so the first file now, or first directory, is called assembly barcode 10. So that's what we just made with Fly. So if we cd or change directory into the assembly barcode 10 file that we just created with Fly, and then we list, we've got a bunch of different files and folders that were created. So when I do an assembly of mitochondrial genome, the first thing that I look at is I'm going to use the cat command. So cat meaning I want to look at the output in a file. So I'm going to cat and I'm going to do assembly underscore info.txt. So this file right here. We're going to look at that. So this file contains information about the genomes that was assembled, or the genome that was assembled. And so if you look here, got sequence name, and you can see it generated two of these things called contigs. So contigs are basically just like assemblies or pieces of DNA, or not pieces of DNA, but fragments that are assembled into these long sections. So they're called contigs. But it could be, potentially that one of these contigs is our full mitochondrial genome. So if we look at contig uh, 4 and then contig 1, there's not really any way just by looking at the names to tell us which one is the mitochondrial genome. So we're going to look at these other columns. So next we've got one called length. So this is the full length of the contig. And if you remember, we want our mitochondrial genomes to be between that 16 and 17 kb. So we see that both of them are within that length. So that doesn't help us narrow anything down. So then we go to the next column called coverage. So coverage is how many reads were used to create that contig. And so usually more coverage means a better, more confident assembly. So we want the one with 72 versus the one with 7. 72 will be the better assembly. And then these other columns we're not going to look at right now. But so we're thinking contig 4 is likely our mitochondrial genome. So the next thing that I would do is I'm going to go and I'm going to cat the assembly.fasta. So if we list, we've got something called assembly.fasta. So that's where our actual sequence of our mitochondrial genome is. So I'll do cat assembly.fasta. So we've got our full sequences of our mitochondrial genome in here. And so this one up here would be contig 1, and this is contig 4. And since we decided by looking at the info that contig 4 is our correct sequence, we're going to just quickly copy this sequence, and we're going to paste it into NCBI's website called BLAST. So we're going to paste that in here. So basically, BLAST is a way to search our sequence and in comparison to all of the sequences that are housed within NCBI. So it will hopefully give us a look into what the correct ID might be and give us some direction here. So this shouldn't take too long. Okay, great. So we've got some uh, some mitochondrial genomes here that are matching to our sequence. And so when I do this, I'll usually first look at the query cover and then also the percent identity. So when these are high, when they're like 95% or above, that means we've got good coverage. So that means our sequence is matching up with this sequence or this sequence here at about 99%. And then percent identity means how similar it is to that sequence. So when you're seeing above 95, you're like, okay, that's very similar. So that's really a good way to increase your confidence. So this first one is Pteranodus sp. So that doesn't really help us get to a species ID. But this next one here is Pteranodus rubiginosus, and it's a mitochondrial genome. And we can see we're at 99% coverage and 96% identity. So that to me is very a good confidence boost to say, okay, maybe we're looking at a Pteranodus rubiginosus. And usually at this point in our study or in our field sequencing, we've seen the bat, we have the measurements, we have the range data. So we're slowly piecing together this puzzle of what it might be. So we've got, yeah, this ID, potentially Pteranodus rubiginosus. However, if you look at some of these other ones, Pteranodus parnellii, Pteranodus personatus, these are also pretty similar, but slightly less similar than this one. So this is 98 and 91% identity. This one's even less, 86% identity. So to me, right now, this one is the most convincing. So the next step in this process would be I would take the mitochondrial genome and I would likely annotate it using the Galaxy Mitos 2. So this is just the Galaxy website with the Mitos 2 tool pulled up. And I'm not going to do this in front of you because it does take a minute, but I did do it beforehand. So here I'm visualizing the mitochondrial genome in a software called Genius. There's also a software called Mega that can work, and Mega is free versus Genius is as a paid software. But you can see now we've got our CO1, so our cytochrome oxidase 1 gene labeled here. So that's what annotation does, is it helps us label which sections are certain genes. And so then you could potentially pull out the CO1 gene or the cytochrome B gene and make a phylogenetic tree with that to confirm what we've seen with BLAST. And so you would do that likely using RexML or another phylogenetic tree construction. But I'll go back into my slides here. And so there's our mitogenome assembly. And then here is a phylogenetic tree that my lab has assembled of this data. So this one indicated with the yellow star is our Pteranodus, or so this is our sequences that we sequenced together. And you can see we've labeled it Pteranodus rubiginosus, and that's because it is grouping with these other Pteranodus rubiginosus, and especially ones near Guyana. So Suriname is close, Brazil, French Guiana, those are all in the Guianan Shield. And it's more closely grouping with those versus the other types of Pteranodus. So that gives us really good confidence that this is what this mystery species was. So our final species ID that we're going with is the Pteranodus rubiginosus. And so this bat is from the family Mormupidae. It's commonly called Wagner's common mustache bat. And it is really interesting because formerly it was considered a subspecies of Pteranodus parnellii. So that's this other one of these other species that was closely related. And it's an insectivore. But one of the really interesting things, and a good reason why we, did this molecular study in the field was we were using this field guide to try to parse out some of the more cryptic species. And in this field guide, it doesn't even have Pteranodus rubiginosus because it was based on data from before this Pteranodus rubiginosus was sort of made into its own species. And so it even says here with the Pteranodus parnellii identification that this is a cryptic species complex, and they're recommending using acoustic or genetic data for the final species ID. So thought that was really interesting, and it's a great confidence booster for why we used our molecular ID here. And so then at the end of these slides, which I think will be available to you guys after the talk, I've put a bunch of resources for learning bioinformatics and this analysis specifically. So all of these first resources are for helping to learn bioinformatics or Linux command line. And then these resources down here, so I've got NCBI Blast, which is the search engine I used to look up our sequence. Barcode of Life is a great database of cytochrome oxidase 1 sequences. NCBI organelle RefSeq, so this is where I got the reference quality level mitochondrial genomes. And then I've also linked the paper that I spoke about that is also linked in the HTML file. And then finally, Anaconda is just a way that you can download software to use. So there's a link to that as well. So I'd like to thank a bunch of people. So the Guyana research team led by Chris Schaefer and Marissa Milstein, couldn't have done this work without them. Our Guyana collaborators, the YY community, Quabana community, and Mahaika communities. I'd like to thank my advisor, Peter Larson, Larson Lab, and specifically my lab mate, Evan Kipp, as well as Doctors Chris Falk and Tiffany Wolf. And then the funding for all this work was provided by University of Minnesota. And I'd also like to thank my co-authors on the paper that helped me to create this bioinformatic pipeline. And again, just to mention that this is based on real stuff, but it's not exactly a representation of, anything that happened specifically. And so I've got my email here. I'd love to take questions if there's any time. And I also want to mention that in the slides that will be published after this, I've included more resources down here and explanations about some of the file formats and types of analyses that we were doing. So that's all.
All right, we have a few minutes for questions. So if anyone has any, please feel free to put them in the chat. If there's something in the talk that you would want to go back to, do remember that this is posted up on YouTube. It'll probably be posted within the next few hours, so you can always go back and watch it again. And we will be posting all of the resources that Lexi has graciously provided for us. They will be posted online as well.
Yeah, thank you all for watching and attending.
We might have
And
a question.
oh, okay, great.
Yeah, I'm too lazy to type it out, so I'll just ask. So I had two questions. So you're looking at mitochondrial DNA. Um, Depending on the species, and like you've shown, this was previously a species complex and could be even more complex than what you've teased out for this one species. But that doesn't always work very well, just using mitochondrial. So have you encountered any issues? I mean, you're using at least probably the site B and CO1. So at
Yeah,
least using two genes is better than a lot of people just using site B. That's my experience.
yes.
Um, so I didn't know if it's just because it's a time saving because you don't want to have to do like whole genome to get the nuclear. Um, can you?
Yeah. Yeah, so you're totally right. With the mitochondrial genome, it's not foolproof and there are certain issues with it, especially when you think about maybe like mitochondrial capture where you're having like hybridization and then, you're coming out with just one mitochondrial genome, even though it is a hybrid. So there are certain issues you need to look out for. In this scenario in the field, we're using mitochondrial genomes, kind of like you said, because it's time saving. We can't really be assembling full genomes to that higher coverage in the field. I mean, we could, but we'd only be doing like one sample in the time that we're in the field. Um, so we're also, combining it with our other metadata, so like the measurements, the morphological data, and then, yeah, typically we're using both genes, if not just the whole mitochondrial genome, to make these phylogenetic trees. And then the other thing about it is like, yeah, they're really complicated, species relationships that we haven't parsed out. There might be, sequences that are labeled on NCBI that are not even correct. But the great thing about it is like we have the genetic data, we can go back to it, we can, add in more sequences as they're created and be able to have that information forever. So yeah, I think hopefully that answers your question.
Yeah. I don't know if you've heard of the BAT1K genome project, but yeah, in case you guys start doing or want to give them samples, that's another thing I was going to ask about. Your samples are lethal, it looked like from your flow chart. Have you tried this using non-lethal samples like rectal swabs,
Yeah,
feces?
yeah, so I've tried it, yeah, with both terminally collected samples as well as non-terminal. So the paper that I was mentioning that I published last year was specifically with fecal samples. So, totally non-terminal samples you can do it with. I haven't tried it with a swab, anal or oral yet, but I'm hoping to be doing that, just in the next couple months. Like, I'm quite sure it will work, but just to make sure. But yeah, you can do it with many different sample types.
Great. Thank you. It was great talk.
Thanks.
All right, well, we are just past 9 o'clock, or well, 9 o'clock for me, 10 o'clock Eastern. So we are going to wrap it up here for the day. But if you want to go back, please feel free to visit the YouTube channel after today. And Lexi provided her email. So if you have any questions, please feel free to contact her. Amazing presentation. Thank you so much for being our speaker today.
Thank you. Yeah, and feel free to email me with any questions.
All right, thanks everybody.
Bye.